Hello everybody and welcome to Unit 14. Today we're going to talk about the blood and tissue protozoans, also known as the hemoflagellates. Today we're going to talk about leishmania, trypanosoma, and plasmodium. Those are the causes of leishmaniasis, Chagas disease, African sleeping sickness, and malaria respectively. As we go throughout the lecture, make sure that you know the intermediate and definitive hosts, the insect vectors, and the diagnostic and infective stages. So here we go. I first want to go over some terms because the life cycles of these hemoflagellates can be very complicated. An intermediate host is a host that harbors the larval or asexual stage. The insect vector is almost always the intermediate host, except for in malaria. Malaria has a backwards life cycle where the human is the intermediate host and the mosquito is the definitive host. But anyway, the definitive host for most of the time harbors the adult or sexual stage, and a human is almost always the definitive host, except for in malaria. The definitive host harbors the sexual stage, and usually that is a human. Trypanosoma and leishmania multiply in the blood, and they all require an arthropod intermediate host, and we're going to talk about those. There's various morphologies that exist in the blood of humans and arthropods, and there's several developmental stages or 4 stages in the life cycle of these organisms. So the life stages in the organisms trypanosoma and leishmania would include the amastogote, the promastogote, the epimastogote, and the trypomastogote. And we're going to talk about all of those stages in upcoming slides. So here we go. The amastogote is an intracellular inclusion that you can see within macrophages of the heart, skin, spleen, and liver. This stage in the life cycle of trypanosoma and leishmania is also known as the leishmanial form. This is an intracellular developmental stage found in the tissues such as the heart. No flagella are seen at this stage. The amastogote literally means no flagella. Okay? So you can see an image there on the right of an amastogote in a smooth muscle tissue, and then above that would be a drawing of the amastogote. And again, this is the first of 4 developmental stages in trypanosoma and leishmania. The second developmental stage often happens inside of the insect host. This is called the promastogote stage. This is also known as the leptomonad form. And again, primarily this form is found in the insect vector. Here we have a single anterior flagella and no undulating membrane can be seen. The third developmental stage for trypanosoma and leishmania would be the epimastogote. This is also known as the crithidial form because it looks like the Crithidia organism. Here we can see in the image we have a short undulating membrane. There is flagella present, and usually this is a transitional stage that occurs inside of the insect host as well. Usually this is where the organism starts to multiply in numbers. The trypomastogote is the 4th developmental stage, and this is called the trypanosomal form. This form can be noted in the peripheral blood, sometimes in the tissues such as lymph node tissue, and even in the spinal fluid. They're very easy to recognize, as you can see the undulating membrane on the organism to the right. Okay, there is flagella present as well, but really what's going to jump out at you is the undulating membrane. Let's first start by talking about the trypanosoma life cycle. And this is trypanosoma that is not cruzi. So Trypana cruzi causes Chagas disease, but this is trypanosoma rhodesiense and gambiense, and basically they are the cause of African sleeping sickness. What happens in this life cycle is a tsetse fly takes a blood meal from the infected person and sucks up trypomastogotes from its host. The tsetse fly here is the intermediate host. The trypomastogotes undergoes an epimastogote stage in the insect gut at this time. The trypomastogotes move to the salivary glands of the insect and then are injected into the next person, and that is the infective stage. Okay, so that is the life cycle of trypanosoma gambiense and rhodesiense. Leishmania is not something widespread in the United States, but there have been cases linked to places in Texas, and also there have been outbreaks in South America. There are three forms of leishmania, and these forms are called the cutaneous form, the mucocutaneous form, and the visceral form. The cutaneous form of leishmania or leishmaniasis affects the skin. The mucocutaneous form affects the mucosa, and the visceral form affects the internal organs like the liver and spleen. These may be causes of lesions and ulcerations known as cauliflower masses, also cartilage and mucosal erosion of the mouth and nasopharynx. Leishmania is found throughout the world in the Mediterranean, Asia, and Africa, and there are 4 species of leishmania, including Leishmania tropica, which causes Old World leishmaniasis, Leishmania brasiliensis, which causes New World leishmaniasis, and Leishmania donovani, which causes visceral dum-dum fever, or this is also known as Kala-Azar, but this is mostly fatal if it's untreated. And then finally we've got Leishmania mexicana, which causes a cutaneous leishmaniasis. Let's take a look at the leishmania life cycle. First, we have a sandfly that takes a blood meal. The sandfly sucks up the amastigotes from an infectious human, dog, fox, or rodent. So here the sandfly is the intermediate host and the insect vector. Promastigotes are injected into the human, and that is the infective stage. Okay, the human is the definitive host. Macrophages engulf promastigotes, and they transform into amastigotes. Amastigotes multiply intracellularly, and that's the diagnostic stage. So in other words, here we can see the amastigotes on a microscopic exam or a diagnostic stage of a skin biopsy or bone marrow aspirate. The sandfly takes a blood meal and ingests the amastigotes. Amastigotes transform into promastigotes inside the fly, inside of the sandfly, and promastigotes multiply by binary fission. Promastigotes then are injected by the fly into the next human host, and that, of course, is the infective stage. Okay? The sandfly is from the family Ceratopogonidae, and the genus, which is very interesting, is called Phlebotomus. I think it's very interesting that the sandfly, which takes blood meals, is called the phlebotomist fly. So here's a picture of the very minute sandfly. It has kind of a goldish color to its hairs, and you can see that it has transparent wings as well. Sorry, some of these pictures can be kind of graphic. The first picture is a cauliflower mass or ulceration, and the second picture down below is erosion of the nasal septum. Diagnosis of leishmaniasis includes skin biopsy exams for amastigotes. We're looking for the amastigotes in a skin biopsy, or we're looking for amastigotes inside of macrophages or inside of a bone marrow aspirate. Let's go ahead and compare Trypanosoma rhodesiense and gambiense. So Trypanosoma rhodesiense causes what's known as East African sleeping sickness. And Trypanosoma gambiense causes West African sleeping sickness. Okay? They both cause sleeping sickness diseases, and they are both transmitted by the tsetse fly. Both have central nervous depression as part of their symptoms. Coma and death are other symptoms and signs as well. The only difference really is that Trypanosoma rhodesiense causes East African sleeping sickness, and Trypanosoma gambiense causes West African sleeping sickness. Let's take a look at their life cycles in the next slide. Let's take a look at the life cycle of Trypanosoma rhodesiense and gambiense. Okay? The tsetse fly takes a blood meal and injects trypomastigotes. That's the infective stage. The tsetse fly is the intermediate host and insect vector. Epimastigotes develop in the fly gut. Trypomastigotes are carried from the bloodstream to other body sites. Trypomastigotes multiply in the human in several places by binary fission and migrate to the blood. The diagnostic stage is when you see trypomastigotes on the peripheral blood smear, in lymph node biopsies, or cerebral spinal fluid. The tsetse fly ingests trypomastigotes from human blood, and the trypomastigotes multiply in the fly gut by binary fission to form epimastigotes. Those epimastigotes then transform into trypomastigotes in the salivary glands of the tsetse fly. Okay, and then what happens is the tsetse fly takes the blood meal and injects those trypomastogotes, and the cycle starts all over again. So here's a picture of the tsetse fly. It is in the order Diptera, and the family is Glossinidae, and the genus is Glossina. And that's very interesting because you can see it has a very large proboscis, and so we do like to say that they are Glossinas. That's the genus, okay? Interestingly enough, the tsetse fly is larviparous and a K-strategist, which means that it only lays a few healthy young larvae. Trypanosoma cruzi is also known as Chagas disease. This causes a weak heart, enlarged lymph nodes, and fever, and may be deadly. The insect vector for this organism is the Triatoma or Reduvidae bug, primarily distributed in Mexico, Central, and South America. Okay, there have been a few cases isolated from Texas and California as well. Let's take a look at the life cycle of Trypanosoma cruzi. So in the life cycle of Trypanosoma cruzi, the Triatoma bug, or the assassin bug, it's otherwise known as the assassin bug, takes a blood meal. Okay, the Triatoma bug is the insect vector and the intermediate host. Okay, this is a hemipteran insect that sucks on plants and also blood. Okay. Trypomastogotes enter the human when triatoma fecal material goes into the bite wound. Trypomastogotes invade various cells and transform into amastogotes. Amastogotes then multiply in the heart tissue or transform into trypomastogotes and go into the bloodstream. That's the diagnostic stage when you can see either trypomastogotes or amastogotes on peripheral blood smears. Okay? The triatoma insect ingests the trypomastogotes, and the epimastogotes form inside that triatoma bug. They multiply and form trypomastogotes inside the triatoma as well. Then trypomastogotes can be passed on again to another previously non-infected human. Okay, so the samples that we look for when trying to diagnose Trypanosoma cruzi would be heart and blood samples where we can see it with right stain. Okay? Usually blood and spinal fluid are examined for Trypanosoma rhodesiense and gambiense, but heart and blood fluids are examined for Trypanosoma cruzi. So here's a picture of the Reduvidae bug, also known as the assassin bug or the Triatoma bug. Okay, this is in the order Hemipteran in the family Reduvidae. Let's go ahead and talk about malaria. Malaria is the most significant infectious disease of all time. There are 550 million people that develop malaria every year, and 1.3 million people die annually from malaria, usually as children of sub-Saharan Africa. There is some indication that Viagra will actually stop malaria from invading the red cells because Viagra actually stiffens the cell wall and it cannot be penetrated by the Plasmodium organism. Okay. Now, there are 5 different types of malaria parasites, including Plasmodium falciparum, that's the most deadly. Plasmodium vivax is the most common, and then we also have Plasmodium malariae and Plasmodium knowlesi, and I think that's all of them. Okay, so let's go ahead and take a look at the malarial parasites. These are sporozoan parasites and have a life cycle that includes sporogeny and schizogeny. Okay, the genus of organism that causes malaria is Plasmodium, and there's 5 species: Vivax, Falciparum, Malariae, Ovale, that's the one I forgot, and Knowlesi. And it is transmitted by the female Anopheles mosquito. The female Anopheles mosquito here is the definitive host, so that's why this life cycle is a little bit different than most other parasites. Some symptoms of malaria would include an enlarged spleen, fever and chills that is very pronounced, intravascular hemolysis, and death. And the malaria organisms take on a very complicated life cycle with two stages. One stage, sexual multiplication or sporogamy, that happens inside of the insect, and then we have another stage, the asexual stage, known as schizogamy, that occurs inside the human. Okay? So let's go ahead and talk about the malaria life cycle in the next coming slides. First, I want to show you a map of the world and where chloroquine-resistant malaria exists and where chloroquine-sensitive malaria exists. You can see it's mostly in the warmer parts of the world. And then up in the top left there, you can see a dipteran, family Culicidae, which is actually the Anopheles mosquito. And Anopheles mosquito actually means useless in Greek, which I think is very interesting. Let's go ahead and take a look at the very complex life cycle of the Plasmodium organism. Like I said, they have a very complex life cycle. The infected mosquito bites a human and injects its sporozoites. That's the infective stage. The sporozoites penetrate human liver cells, and then the sporozoites become cryptozoites, then schizonts and merozoites. The merozoites are released and invade RBCs. They develop into mature trophozoites or ring forms. Some merozoites become gametes or gametocytes. Gametocytes would include microgametes, which are essentially male gametes, and macrogametes, which are essentially female gametes. But these gametocytes are sucked up by other mosquitoes and passed on. The gametocytes are sucked up by another mosquito, and then an ookinete and oocyst are made from the conjunction of macro and microgametes. They rupture and release more sporozoites. People with sickle cell trait are somewhat protected from malaria. Don't forget that schizogamy is an asexual stage that happens in the human, and sporogamy is the sexual stage that happens in the mosquito. There are 4 stages of malaria seen on peripheral smear exams. What you're likely to see most is the ring form. This is also known as the immature trophozoite. Then we have the trophozoite form, which is enlarged and more mature. We've got the schizont and the gametocyte. Take a look at the handout that I gave you in your packet so you can see the chromatin dot and the ring forms, the troph forms, the schizont forms, and the gametocyte forms. Only certain stages of the Plasmodium development will be seen in the peripheral blood depending on the species. Thick and thin blood smears should be examined with right stain. Currently, there is no serological test available to diagnose malaria. Take a look at the ring stage. This is from Merozoic or Merozoite development, and what you can see in the ring stage is a dot of chromatin with a trophozoite ring around the dark chromatin dot. This is a common form seen in peripheral blood smears. Got to be careful though, because it almost looks like a platelet laying on top of the red cell. This is the trophozoite stage, and they appear thicker and more compact, the ring forms do. The more mature stage, where the ring appears to be enlarged, and it contains a single red nucleus and also Schiffner's granules, which may be seen in the red cell, but not usually seen with Plasmodium falciparum species. The nature of the Schiffner's dots is actually uncertain. In the schizont stage, the nucleus splits into several daughter cells. These daughter cells appear as an arrangement of oval nuclei. They're about to become merozoites, and I will tell you the schizont stage is not often seen in peripheral blood, but 8 to 24 mature compact trophozoites, dark and tightly clumped into a mass, is what you're going to see. The merozoites, it's the merozoites that actually attack healthy red cells, not the schizonts. And usually there's 6 to 12 merozoites inside of each red cell. In malaria, the only stage seen extracellularly is the gametocyte stage, okay? So merozoites break out of the red cell and are seen externally. Large crescent-shaped gametocytes are especially noted with Plasmodium falciparum, and this is when the patient has extreme chills and extreme fevers. Pretty much when the merozoites break out of the red cell. Here we've got a ring form on the upper left. a schizont form on the bottom left, a trophozoite on the upper right, and a gametocyte on the bottom right. Okay. Now, malaria can be treated with chloroquine drugs, but you never really know if it's fully cured. It can actually hibernate and still be viable in any form, anytime from the liver. If you like vintage war time videos about things like malaria, here is a link that you can copy and paste into your browser. It's about a 9-minute Disney video talking about how to prevent getting malaria. Babesia causes Texas cattle fever. They have similar symptoms as seen in malaria, and there is a worldwide distribution of Babesia. Ticks are the definitive host. There's actually over 100 species of Babesia, and a person may be infected with 2 or 3 Babesia species at the same time. Ticks are the definitive host. Tick bites an infected rodent, and then tick is now the intermediate host. Okay? Tropes can be seen outside of the human red cells, and this never happens with malaria. The only time you're going to see something outside of the RBC in malaria is in the case of gametocytes, but the ring stage is never seen outside of the red cell in malaria. Okay? Or you can see the ring cell outside of the you can see the ring form outside of the red cell in Babesia. Okay? You can treat Babesia with clindamycin or the quinine drugs. And let's take a look at the Maltese cross formation of Babesia versus the ring form of malaria. So the 3 images, the 2 top images and the bottom left images is all pictures of the Maltese cross formation of the troph form of the Babesia inside of the red cell. You can see that the troph form is a little bit more elongated, and they will make Maltese cross formations. If you compare malaria troph rings to the image on the bottom right, you can see they are more circular, the ring forms. So that's it. That's everything you ever wanted to know about the hemoflagellates. Today we talked about leishmania, trypanosoma, and plasmodium. If you have any questions, please reach out.